Hi, welcome to this lecture on environmental adaptation of animals. On these videos, we will consider more on the metabolism. So how we can produce ATP for our cells. And what about then, if, for example, in the exercise, is the ATP production enough for the ATP consumption? And let's start with the crayfish. About 25% of the body mass is the tail muscle. And it's needed for the backyard swimming, and usually if you have a crab that is moving backyard, it's escaping or hiding. It can do it very fast, and there's a variation in the oxygen consumption. So in this crayfish escaping, it will have a burst of exercises, maybe 15 or 30 flips, after which it's exhausted. But at the same time, most probably it's also hidden from the better predators. So it's like running as fast as you can. You can't do it for ages, only for few seconds or few tens of seconds, etc. But then the other way to move is to make a sustainable exercise where there's ATP, enough ATP for all the energy needs. And okay, in that case, you can run for a longer time or move a longer time. And in every cell, the ATP production and the consumption should be in some kind of balance, at least in long run, because ATP cannot be transported from one cell to another. So there's an energy shuttle. ADP can be from ADP, you can produce ATP if you have enough energy. But then from ATP, you can cut the last phosphate to produce ADP and then release the energy. And of course, the ATP can produce with two ways. Either by using aerobic metabolism, so meaning oxygen, or anaerobic metabolism without oxygen. So let's have a short review of aerobic catabolism. Okay, we all know that the aerobic conditions, we have glycolysis, citric acid cycle, electron transport chain, and oxidative phosphorylation. And it all starts with the glycolysis. That is something that you should already know quite well. And it's happening in the cytoplasm, and it has three main characters. Glucose is converted to 2-pyruvate. And there are two molecules of NAD that is reduced to NADH per molecule of glucose. And it happens there in the middle, and it happens only once in this chain, but because glucose is cleaved, or actually fructose 1,6-diphosphate is cleaved to two sister molecules that can be converted to each other. So that's why we have the end of this road twice. And two molecules of ATP is used. So the glucose is phosphorylated, and then later on the fructose 6-phosphate is again phosphorylated. So you increase the energy of these molecules. And later on, you will have two reactions where ATP is produced, but it happens two times, so meaning that, okay, you have the net yield is 2 ATP per glucose, because 2 were also used in the beginning. Okay, if we have oxygen available, then the rest of this catabolism happens in the mitochondria. So the pyruvate is transported in the mitochondria and modulated in smaller parts. And this happens by citric acid cycle, citrate cycle, Krebs cycle. It has many names. And what happens there is that the glucose is cut to produce 6 carbon dioxide. There is 2 carbon dioxide on the right hand side and 1 there on the top. And because this cycle is going 2 times per each glucose, so 3 plus 3 is 6. And at the same time, 8 molecules of NADH is produced. 1 on the top, 2 on the right hand side, 1 on the left hand side. And 2 FADH is produced also, that happens there on the left hand side. So you get hydrogens somewhere, but actually so far you have not got any energy. And in this citric acid cycle, ATP is produced single time on this round, and because it goes two rounds per glucose, so actually that's why we get 2 ATP. While in this figure it's actually made GTP, but this GTP is giving the phosphate for ADP to produce ATP. And what happens for this NADH and FADH? They are giving the hydrogens on the electron chain. So they give the hydrogens for this electron transport chain, and it goes like this. So the NADH is converted to NAD, and at the same time it's releasing electrodes. As well as FADH is converted to FAD and releasing electrodes. So it's releasing protons and electrodes. And these protons are pumped in the intramembranous cell space with the energy of these electrodes. So in this electron transport chain, we have complex 1, NADH dehydrogenase, that is taking something from the NADH. And then we have complex 2, succinate dehydrogenase, that is affected by FADH2. So FADH2 is giving the protons there, or at least electrodes. And these electrodes, in the end, they are bending there on the right hand side to oxygen to form water. And they go through this electron transport chain, and they give energy for these membrane proteins. And how they are giving the energy is that, then these membrane proteins are pumping protons in this intermembrane space. And what happens next? We have a proton gradient. So when they are pumped out from the mitochondrial matrix, they form an electrochemical gradient. So they try to get back. And because there is ATP synthase, the flux can be modulated to chemical energy. So if there is 4 protons going back in the mitochondrial matrix, it will form about 1 ATP molecule. But at the same time, there is a channel for protons. So this kind of uncoupling protein that is lowering this ATP production. And it can be lowering the ATP production even to 0. So all the protons that are pumped out are just flowing back. And why we have this kind of uncoupling? Well, the reduction of NADH move 10 protons and FADH2 move 6 protons in the membrane space. And this is calculated quite often that, what is the how much protons versus oxygen is used. So what is the proton oxygen ratio? And if we get 10 NADH or 2 FADH, then we can get a lot of protons in this intermembrane space. And when they are flowing back, for every ATP we use 4 protons, and that's why from this process we can get 28 ATP from 10 NADH and 2 FADH2. And the uncoupling in the uncoupled cell, the ATP production happens only in glycolysis and citrate cycle, meaning that, okay, only 4 ATP can be produced. But the good point is that, okay, then the proton flow is turned to heat. And this is the major mechanism, increasing the metabolic heat in mammals in cold season. So that's what happens in the Finland in the winter time. This is the way how the animals are increasing the heat production to prevent to get cold. But actually these calculations are a little bit nonsense, because there's always some leak in this ATP synthase. So there's always some channels open, and in some tissues even 20% of the pumped protons are just leaking back. And furthermore, it's been speculated also that the glycolysis, because it's producing this NADH, the transportation to mitochondria may be limiting also the energy product on that side. So that's why it's definitely less than 32 ATP per 1 glucose. Okay, what happens then if we don't have oxygen available? Okay, if we don't have in this process oxygen, the electrodes entering in the electron chain they cannot be discharged in the reduction of oxygen. And therefore, the electrodes are accumulating in this electron chain components and stop the work. And at the same time, because there's no one taking the protons away from NADH or FADH2, there is a redox balance problem. And this may stop the energy production completely if NADH from the glycolysis cannot be oxidized. And furthermore, if we have a little bit oxygen but not enough, the problem also can be that we will actually get too much reactive oxygen species. This happens all the time in the mitochondria, but surprise, surprise, we have some kind of protection mechanisms. So sometimes there's when we are giving the electrodes, we can give wrong number, and these can cause the reactive oxygen species. But we have superoxidized dismutase to produce hydrogen peroxide that we can catalyze to perform oxygen and water. But especially in case when we have lack of oxygen, we can have several damages, for example in neurons, in the areas where we have little bit oxygen, or when we can get the circulation refluxing again, then we can get high damages. What about then in anaerobic metabolism? Okay, you all know that, okay, the brains cannot produce enough energy without oxygen. But still, in some tissues, we can run as fast as we can, and we know that, okay, it will go anaerobic. So in some cases, the energy can be produced even with anaerobic glycolysis. And this regards the recycling of NADH. So when producing from glucose to pyruvate, we produce at the same time NADH. And it must be recycled with lactate dehydrogenase when we are producing lactate acid. And this reaction is of course quite ineffective. So we can only produce 2 ATP per glucose. And the problem also is that we can't use fat, we can't use proteins in this kind of synthesis. We only can use sugar, meaning the glucose. And the problem also exists that, okay, lactic acid is an acid. And if we have acids, we lower the pH. It's a weak acid, so nothing to compare these sulfur acids, etc. But still, when it's accumulating in anaerobic cells, it lowers the pH. And therefore, the animals that want to tolerate long-lasting anoxia must find either way to modulate the lactate acid, release it from the cells, or find another way to produce the energy without the oxygen. And this is a little bit old stuff, but as you can see, that even among anoxia-tolerant fish, like carp, tilapia, or even goldfish, that is actually quite much the same as our Crucian carp, the pH is dropped drastically during the first 10 minutes or 1 hour of time when there is no oxygen available. So the pH is decreased because now there is more lactate. Okay, but then suppose that, okay, I was running as fast as I can to reach the basketball, but so what to do with the lactate afterwards? We can metabolize it back to pyruvate if we have oxygen. So it's not a catastrophe to have the lactate in the skeletal tissues. And after getting it in pyruvate acid, we can then turn it either to by gluconeogenesis to glucose or even the glycogen, or use it for energy production. So it's just the storage that we can use. And actually, when it's been tested with radioactive carbon isotope, it has been demonstrated that this lactate acid can be also carried via the circulation and metabolized in other tissues, even when the lactate production is still continuing. So we can a little bit help it by moving it in other places, but it's still, in case of long-lasting hypoxia, that's not the good way to do. Okay, we can have long and short-term energy use. So we started with the crayfish in the beginning, that okay, we can walk slowly or escape fast. So in the steady-state mechanism of ATP production, as much ATP is produced with the oxidative catabolism as it's used. And there are no chemical byproducts that are accumulated because they are used as fast as they are made. And this kind of metabolism can continue actually forever. But then in comparison, the non-steady state mechanism of ATP production, it's limited. The production, the product will, the supply can be limited, or the product will accumulate, causing some self-limited processes. And there can be some alterations in cells causing the continuing physiological impossible. If you ever have run as fast as you can, as long as you can't do it forever. And, well, what happens there? It's accumulation of lactic acid, acidification, but also other changes that we will discuss greatly on the next videos. One of the things that happens that or can be used is phosphogens. So we have creatine phosphate in vertebrates or arginine phosphate in invertebrates. They are batteries. They are able to store phosphate. And in case there is need for ATP, they can give the phosphate to ADP. And it goes on or works on the mass action law. So if there is too much ADP, okay, then the creatine phosphate is giving the phosphate to produce ATP. But if there is no ADP at all, then this reaction is not working. And that's why when we are running fast, as long as we can, the ATP that is produced first in the mitochondria, now it can go for 3 or even 6 times from the contractility properties in the cells, and then back on the creatine phosphate, and then back on the contraction, etc. So it can go this kind of cycle even for 6 times. But still, it gives only temporary help for the energy needs. It's essential for our living, but it's not the way you can live forever without oxygen. And another way, besides these phosphagens, is also that, okay, we have in the muscles, we have the myoglobin, that is almost the same looking word like hemoglobin, but now it's in the muscles, and it can bind oxygen. So we have oxygen storages. But even then, both these phosphagens and myoglobin, they are only temporary help. And even in these animals that are able to live quite a long time without oxygen, as well as we saw that, okay, over here on the right hand side, the pH was decreasing because there's more lactate. Also the phosphocreatine levels are decreased. For example, in the carb, within half an hour by 90%. So now we have 4 different ways to produce ATP for the cells. We can have aerobic catabolism that we already know quite well. Then we can have these anaerobic glycolysis, except in the brain. Then we can have these phosphagens, like creatine phosphate. And then we can have these oxygen storages like myoglobin. And all except the aerobic catalysts are working only for short period. So they are not in the steady state at all. And that affects the total yield per episode in moles is completely different. So in aerobic metabolism, during the marathon, the ATP yield is very large. And of course, during the lifetime, it's maybe 4 million. And then in the anaerobic glycolysis, it's still something. So we can have little bit ATP produced because it's quite long term happening. But then these batteries, either we are having in the battery phosphate, like in these phosphagens, or we can have a battery of oxygen in these oxygen storages. They are very tiny. So there's only limited number of ATP that we can produce during these episodes. Then the difference is also that the rate of acceleration, how fast we can make it faster or more efficient. You can see that the only thing that happens slowly is now the aerobic catabolism, because it's already working quite often as fast as it can. So it's a very slow process to have more mitochondria in your cells. It doesn't happen just in a few minutes. But these anaerobic glycolysis or phosphagen or oxygen storages, these are the things that we can do quite fast. And also the peak rate of ATP production, so how many micromole of ATP per gram per minute, is completely different. So we can have maybe 30 micromole of ATP with glycogen and then 20 with fatty acids. But it's still quite low to compare these other ways. So with fast glycolysis, we can have 2 times more ATP in a short term. Or by releasing the phosphate from these phosphagens, it's enormous. And even the oxygen storages can be producing a huge number of ATP for a very short time. And also the difference is that, okay, what happens after the use? Okay, the aerobic catabolism, because it's steady state, nothing happens. And the phosphogens, these bacteria, phosphogens and oxygen storages, they can be reloaded quite fast. But the difference is in the glycolysis, because now we are accumulating lactate. The return is happening quite slow. Thank you.